So I'm going to talk. Sorry, a little bit of biochemistry at the start, and this will be relevant for some of the aspects of the talk later. But DNA basically is where our genes are. DNA is transcribed into RNA that carries the message to be then translated into a protein, and then proteins will provide function for cells. Now, the asterisk on this is that DNA, RNA, and protein all have functional capacity, but for the purposes of the talk, this is how I'll present it. What happens is proteins are made in chains, and then they fold into three-dimensional conformations that then have activity based on ability to interlock with other molecules. And so I'm using clay. I think it's a better example than last year's, but so a normally folded, normal three-dimensional structure of a protein will have a normal function. Proteins can become altered in their shape, they can be modified chemically, and they can actually clump together. So the clay protein here, for example, clumps into a nasty-looking clump of clay with, I guess, grass in it, and so it's not going to do what it was supposed to do. And so clumped proteins can cause disease states. Clumped alpha-synuclein protein in particular has been shown in many different models to be toxic to cells, and there is a lot of data supporting that toxicity from this clumped alpha-synuclein protein can forward the process of Parkinson's disease. Now, I'm going to take a step back. Everything that I'm telling you today, everything does have a little asterisk on it. Not everything I'm going to tell you has been proven. A lot of it is theory based on a lot of good data, but there's a lot of work to do yet. But what I'm going to do is I'm going to kind of trim the fat and present it in a way that fits a story that is very, very plausible and supported by science. So you could poke a hole in almost everything I say today, but for the sake of discussion, take it for what I'm saying, and don't don't take it any farther. So normal proteins like alpha-synuclein can become clumped together, and clumped alpha-synuclein is actually the main component of the Lewy body. Dr. Lewy, 100 or so years ago, saw these little protein nuggets in cells, and back in those days, everything that you saw got your name on it, so it's a Lewy body, and this is what it looks like under a microscope. We'll talk a little bit more about that later, but that's the clump protein. There's a lot of evidence, particularly over the last decade, that clumped alpha-synuclein will actually spread through the brain. All right, so I'm up there, so I don't need to point myself out. Whoops, wrong one. There we go. So this theory has been, this goes back 20 years now, but there's evidence that the alpha-synuclein clumping pathology starts kind of in the base of the brainstem here, but also in the olfactory smell area over here. And then as time goes on, the pathology tends to spread up higher into the brainstem. The areas that started become more pathologically involved, and then as disease progresses, may even spread farther and farther into the brain on a whole. But again, the areas that started are still accumulating the pathology. And that has been a kind of a major impetus for a lot of work that's been done over the last decade. There's also a lot of evidence in more recent years that this clumped alpha-synuclein can be found in the gut and may traffic through a nerve up to the brain stem. So there's brain Parkinson's, gut-brain Parkinson's, and this is something that's still being worked on extensively. There are human diseases that actually work this way, prion diseases like mad cow disease from maybe 3 decades ago. People eating tainted beef in England got these proteins in here. They went into the brain and spread through the brain and caused a lot of damage. So there is a human pathology correlate to that in terms of other disorders of the brain. The brain's not dumb, okay? The brain, when these things happen, the brain works really hard to protect itself. But how does it do it? So the brain, or the cells will make proteins. They'll fold into their shapes to be active. If things happen to chemically modify or alter the shape, the cells will have mechanisms to try to keep that from happening, try to hold them in their shape so that they keep functioning. If a protein becomes abnormally folded or modified in some way, the cells have very robust ways to get rid of the abnormal protein. So there are lysosomes within the cells, which are basically trash cans with acid in them, and you can acid digest the clumped proteins. There are proteasomes within the cells that are like paper shredders, and you can actually shred up the proteins to get rid of them. And then there are mechanisms, and this is a little bit more controversial perhaps, of maybe taking these proteins and putting them in the closet. And this is the way I think of the Lewy body. You got these abnormal proteins, let's take them and put them somewhere so that the cell can still function. There's a lot of talk, is the Lewy body pathological itself, or is it protecting the cell? What I've always thought is when we look at someone's brain after they've passed away and they've had Parkinson's for maybe 40 years, we're looking at cells that were still alive at the time of the person's death that have Lewy bodies. So the cells that have already passed away are gone, we don't see them, and those are really the cells that we want to know what was happening, and then these cells, what did they do well enough to survive the whole course? So I think that the put it in the closet is maybe tenable. Anyway, that's my thought. So there are a lot of hot topics going on. A hot topic, obviously, is alpha-synuclein. I wouldn't have presented it this way if it wasn't a major topic these days. So targeting this clumping process is in various phases of clinical trials at lots of levels of the pathway. So molecules are being used to try to reduce production of alpha-synuclein. There's a clinical trial at Ohio State that is using a molecule for that, and actually, albuterol or propranolol? I think it's propranolol. Albuterol, sorry, inhaler medication can actually reduce production. It doesn't mean everyone should go out and get inhalers, but there's some basic science on it. There are small molecule approaches to trying to block the clumping and aggregation process. There are less of these, more kind of basic science level small molecules being used to see if we can block the uptake of alpha-synuclein as it tries to spread from cell to cell. A little bit more along the line in the pathway, active immunization, so vaccinations. There are scientists that are actually giving vaccinations like we got for COVID, like we get for flu, to put abnormally folded alpha-synuclein in, or at least parts of it. Our immune system then generates antibodies against it to try to eradicate it. That's early stage, primarily kind of a safety tolerability level. And then there's also passive immunization, which is actually infusing antibodies. And this is done throughout medicine. putting in very specific antibodies that will go for and try to remove or alter the function of particular proteins. So there are a few kind of early phase studies looking at actually putting in antibodies to target these clumps to see if we can reduce the clumps. And this is an approach that's actually being used in Alzheimer's right now. Lecanemab is an FDA-approved approach to infusion against the amyloid plaques that build up in Alzheimer's disease. So Parkinson's is a little bit behind there, but people are working on that approach. There are also small molecule approaches to trying to beef up the activity of these cell protective processes to try to make lysosomes work better, to try to make proteasomes work better. Some of the pathologies that are associated with Parkinson's disease actually work to reduce the activities of these cell protective mechanisms. So trying to beef them up pharmacologically is a pharmacological or trial approach. Why don't we have a cure yet? I'm asked this a lot. There are a lot of reasons for that. One of the reasons that really comes to mind is the primary driving pathology of Parkinson's disease is in these deep cells, the substantia nigra, dark substance. It's actually melanin, like when we get a tan, that is built up in these cells. These are, this is a brain, you've got a skull, this is really deep in the upper brain stem. We can't go in and biopsy this. We can't stick tubes in to collect biochemical samples from this. If there's a dermatologist in the room, I'm sorry, but we're very jealous of you. What is that, doc? Well, I'm not really sure. Let me take a piece of it. We'll grow it. We have quick access to the skin, and we can really study that very well. Parkinson's, it's very, very tough to get to the tissue, and particularly what's happening in the early stages. That's what we'd really like to know in these deep brain cells. We just don't have the access that we'd like to have. So one of the transitions past decade, half decade, has been to really start to focus on the genetics of Parkinson's disease. So yeah, these are deep brain cells that we can't get to understand, but we've got genetic pathways that cause Parkinson's, so what's happening there? And maybe we don't need we don't have to rely on sampling those cells. Again, genetics is coming primarily from the DNA within that equation, and there are a number of genes that, when mutated, will cause Parkinson's disease to develop. There are many more than this that will actually cause Parkinson's disease, and then there's a large group of genes that, when mutated, will increase the risk for Parkinson's but not actually cause it. And so we talk about sort of a nature-nurture balance of genetic susceptibility towards a disorder, but then environmental exposures, what have you, that will kind of work together to actually cause the disease. But there are a variety of genes that, when mutated, will actually cause Parkinson's disease to develop. The work that's ongoing now is to try to understand the biochemistry of these genes, not only in terms of what do they do normally when they're normally folded, but when they become altered in some way, what happens? What's the pathway that they're using to cause Parkinson's to develop? And so this is I hope it is this way, but I don't know if the pathways, they certainly do intersect. Alpha-synuclein I've been talking about, glucocerebrosidase, PINK1, LRRK2, Parkin are all very well known, very well established and testable sources of Parkinson's disease. And a lot of these genes actually work on the proteasome process that I was talking about, or the lysosome process, so the process of breaking down the abnormally folded, abnormally clumped disease-causing proteins, alpha-synuclein, when it is doubled or tripled, the mutations will double or triple the production. And when that happens, pathology develops and Lewy bodies develop. So again, a lot of evidence that alpha-synuclein pathology is a driving force. What the scientists are working on is how do these, what happens, how do the pathways intersect, and can we use the intersections to then come up with treatments? So for a gene, let's say we can identify what this gene does, we can test for it, and we learn about the biochemistry of the problem. We develop a molecule or viral approach to correct that function. Then for anybody who has GBA mutation Parkinson disease, we can give them that medication and stop it in its tracks. I think ultimately what we would really like to have is, do we have a kind of a common convergence point in these pathways where we can target that? So it doesn't matter what genetic mutation a person has, and maybe even what we call sporadic Parkinson's, where one of these mutations isn't found, maybe it's something else, but maybe by understanding the genetics and the biochemistry of the genetics, we can find a common pathway point that we can treat to just make Parkinson's stop in its tracks, or maybe even not develop. So that work is ongoing, and that's a lot of scientists are focusing on that. The Parkinson Foundation, and many of you know, I give the information out in my clinic, has a PD generation project where for free, they will actually test by a little oral swabby that you can send off to Indiana University, test for 7 of these genes. A few of the people that I work with have actually found that they have GBA or other mutations. The Michael J. Fox Foundation also is working on this in a very large organized approach called the Parkinson's Progression Markers Initiative. They take a lot of samples, test for a lot of things, including the genetics. And as I said, there are a lot of people working on this. So that has really been kind of a transition over the last decade to really focus on the genetics. Biomarkers are a hot topic. So in other words, molecules that are in our blood, in our fluids, in our tissues that might be altered in Parkinson's disease that will allow by a test signal that Parkinson's disease is ongoing. One of the important uses for biomarkers is going to be, how do we measure disease progression? We don't really have a good way to measure it right now, believe it or not. We have scales. People who are working with me in the Sparks trial will sit down with me every 3 months, and I'll ask them questions and I'll examine them, and I give them scores based on symptoms and then signs in the exam, and they come up with the score, and that is really kind of the state of the art for measuring disease progression. But if you go on a medication, your score will get better. And does that really measure what's happening in the brain? So we really need better ways to show that the treatments that we're giving are actually slowing the pathology. And then, even more important down the line, how can we measure that our intervention is actually slowing disease progression? So we really need to find ways outside of just symptom scales to assess what's happening in the brain. So this is a dopamine transporter scan, or DAT scan. We do not get these very often, so if you haven't had a DAT scan, don't worry about it. It isn't very clinically helpful, so a number of the things I will talk about you do not need to have. I will let you know. I think everyone in the room has heard many, many times the dopamine story. dopamine cells in the brain become dysfunctional and start to die over time. And they are the driving force behind the slowness of movement, the tightness of muscle tone, and the tremor that are the clinical definition of Parkinson's disease. So a normal scan, so what's basically happening here is it's like if I put the cells send processes to this area of the brain that's collectively called the striatum, and we can give a radioactive finger polish, finger paint, so to speak, to see how many fingers of those dopamine cells are still alive and make it there. So on your left, which is also my left, sorry, it's early for me too, is a normal scan. It's called a bi-comma. You can see commas there. On the right side is an abnormal scan. So this person has lost a large number of those dopamine fingertips, so to speak. So, even I as a neurologist can look at this scan and see that there's been loss of dopamine cells. This is not a quantifiable type of scan per se, and you can see it's kind of fuzzy, so it's not 100%. And there are people in the early stage of Parkinson's disease who can still have a normal scan. So I don't get it that often. It doesn't really help me to make a typical diagnosis. But this is a biomarker. Okay, we can get a scan and we can look and see that a person has lost cells if they have lost enough cells for this scan to be interpretable. So another biomarker I've already been talking about a little bit, genes. So, we do a genetic test to see if a gene is mutated. That is a potential biomarker. RNA, again, RNA is made off of DNA and then is used to make a protein, taking basically the signal from the DNA into a protein. Over the last year or two, scientists have produced RNA profiles of saliva, nasal washings, and blood, where there are groups of RNAs present in healthy, non-Parkinson individuals, and then overlapping groups, but then other groups of RNAs that are more associated with Parkinson's disease. So maybe at some point we can do a blood test, look at the RNA population, and be able to tell someone is developing Parkinson's disease. And then certainly in terms of proteins, we can do blood tests and other fluid tests to look at proteins that may be differentially expressed or altered in Parkinson's compared to a non-Parkinson's sample. And no surprise, abnormally folded, clumped, aggregated alpha-synuclein is a major biomarker target right now. A study came out about this time last year looking at spinal fluid, looking at alpha-synuclein aggregation. And there are two tests now, on your left, a spinal tap, and on the right, a skin biopsy that can be used to look for this abnormally folded protein. Please do not worry. I am not going to recommend spinal taps for anybody, and I'm also not going to recommend skin biopsies, and I'll tell you why. It's a really big step forward, however. The spinal fluid analysis showed that in persons who had clinical Parkinson's disease by the examination, stiffness, slowness, tremor, a positive dopamine transporter scan like I just showed you, and either smell loss, which is a early sign, and or what's called REM behavior disorder, REM, R-E-M, rapid eye movement sleep, where we dream. In Parkinson's disease, with the aging process, with a number of medications that activate us, we can have a little bit of an overlap between dream sleep and wakefulness. So instead of being in the normal state when we're dreaming, we're actually paralyzed, except for breathing, which is smart, and for gastrointestinal, which I guess is smart. But at least the breathing part's pretty smart, because otherwise we'd be in trouble. So in the REM behavior state, people will actually be able to act out or yell out in response to the dreams. So under those conditions, the spinal tap, looking for this abnormally folded alpha-synuclein, 99% of the time was positive. So there were a few people with Parkinson's, as far as we could tell, that did not have this finding. Some of the genetic forms are not as focused on alpha-synuclein, and it was probably their influence. But it's a pretty strong positive test to support the diagnosis. Interestingly, there was a group of about 60 people with what we call prodromal Parkinson's, so they had smell changes and or the REM behavior disorder, did not have clinical Parkinson disease in terms of stiffness, slowness, rigidity. 85% of those people had a positive spinal fluid test, but only 30% had a positive dopamine transporter scan. So the idea is maybe well before the clinical onset, and by the way, we think Parkinson's is going on in the brain for 5, 10, 15, maybe 20 years before a person develops tremor, stiffness, slowness. So the idea is we can collect a biological sample pre-symptomatically, diagnose even before the dopamine transporter scan is positive. And again, I've seen a few people who I was sure had Parkinson's who had a kind of equivocal dopamine transporter scan. However, time out. A number of people, about 8% of people with no symptoms whatsoever, even the smell, REM behavior, were positive. So they're either false positives or these are people really early where Parkinson's is brewing. So that still remains to be identified or to be sorted out. Skin biopsy looks a little bit differently at the alpha-synuclein, not quite as robustly, and the data aren't quite as good. The point is, these are ideas for the future. We don't need these tests to confirm a diagnosis now. And someone having alpha-synuclein pathology, unfortunately, today doesn't change how we approach things because I don't yet have a cure or disease-modifying therapy to offer. Whether this is positive or negative, if we think you have Parkinson's disease, we're going to offer you the treatments that we offer. Now, the goal, of course, is can this just be a blood test someday? I don't usually look this happy when I'm getting my blood drawn, but I decided to make it a positive experience for the morning. But rather than pulling out spinal fluid, chopping off skin, if we could just get some blood. And there was a paper that came out within the last year using the same technology for the spinal fluid. It was not as robust, but it did show a very high percentage of people with Parkinson's disease that had the alpha-synuclein clumped in the blood. So I think a blood test is not too far away. The Parkinson Progression Markers Initiative by Michael J. Fox is looking at all of these. They take skin, they take blood, they take saliva, they take spinal fluid, they do dopamine. They're doing all of this, genetic, and the whole idea is to collect thousands of people's worth of data and follow them over time in terms of the biological samples and in terms of symptoms to really understand the base pathology and progression of Parkinson's with the idea of being able to diagnose early. So I forgot that this was my next, but I set myself up perfectly, and maybe that was by design when I was putting the talk together, but early diagnosis is actually a really, really hot topic. It's been for a long time, but it's starting to become much more formed. And so I've already brought up the term prodrome as opposed to Parkinson's disease diagnosis itself. And so the prodrome, as I said, we really do think that pathology is going on in the brain, lower brainstem olfactory areas for smell, well before we can make a clinical diagnosis by tremor, stiffness, slowness, mobility changes. The prodrome, with that blue, okay, blue's better up there than over here. The two major parts of the prodrome, so pre-diagnostic, pre-tremor, stiffness, slowness part of Parkinson's disease is the REM behavior disorder and the smell changes. People will lose their smell or have significantly reduced altered smell and develop vivid dreams. I'm told commonly I'm being chased, attacked, pursued. That's number 1, number 2, number 3, and then number 4 is I'm in some weird version of my old workplace or my old school. So that's what I hear. But by the data, the smell changes are really kind of the primary. Now, I lost my smell with COVID, so, and I do have, I'm getting vivid dreams, so I don't know. maybe I'm brewing too, I don't know. But if there are family members in the audience who lost their smell with COVID, that's a common cause, so don't freak out, okay? But the deep areas of the brain that I was showing up there also, in addition to modulating mobility, also modulate mood. So a lot of people in the years ahead of developing tremor, stiffness, and slowness will notice I'm a little more anxious or prone to anxiety than I used to be. I'm a little more prone to being depressed. I have people say I'm prone to getting pissed off. Fortunately, the dopamine replacement medications often calm that down. Constipation, the gut slows down just like the body slows down, but the water absorption rate doesn't change. So the stool gets a little dry and hard to pass. Vivid dreams, and then biomarkers of the prodrome, alpha-synuclein buildup in the spinal fluid. And then we diagnose Parkinson's disease currently by the presence of bradykinesia, which is slow, reduced amplitude movement, so both components have to be present, and tremor and or rigidity of muscle tone. So that's the clinical diagnosis. But ultimately, what scientists are trying to do is come up with clinical signs and symptoms, prodromal signs, symptoms, and genetic protein RNA biomarkers to make a diagnosis a little bit more firm. The goal, of course, ultimately is pre-symptomatic diagnosis. Why? I think we all know why. We can treat it early, and hopefully one of these days our medical students will be taught what Parkinson's was. They have to remember it for the test. Why do I need to know this? I'll never see it, because you'll go to your primary care doctor, you'll get your blood count, your metabolic panel, your liver panel, your cholesterol, your Parkinson's panel, your Alzheimer's panel, and you look for all of these things. Oh boy, you've got Alzheimer's brewing, or you've got Parkinson's brewing, and we know how to treat it now because we've figured out those pathways, so we'll put you on, and somebody will come up with a fancy name, the TV commercials with all these. I swear there's a college major to come up with the names for these new drugs. Someone will come up with a cool name, but we'll put people on it, and it will just stop the process, and people will never develop it. That's really where this is all going. Exactly. So good luck to the scientists that are doing this. One more hot topic, the gut microbiome. As I said before, there is evidence that these clumped alpha-synuclein molecules are actually in the gut, and there is thought and evidence that these alpha-synuclein clumped particles can actually spread via nerves up into the brain stem. And so a question is, does Parkinson's disease in some people actually start in the gut and then spread to the brain and then spread through the brain? There have been studies, and again, I'm not going to do this to anybody, so don't worry. There have been studies where surgeons have actually gone in and cut this nerve to stop the progression. It hasn't really worked because already going on up there. So we're not going to cut anybody's nerves. But it's a big area of science nowadays, and it's interesting. We have tons and tons of bacteria, viruses in our intestinal systems that help us to digest foods and reduce inflammation. There are a lot of helpful biological processes that happen because of all this stuff in our gut. There have been a number of studies showing it's called gut flora, the the mixes of different types of bacteria and viruses in a healthy gut, in an unhealthy gut, whatever that means, and even in Parkinson's disease. So scientists are working out, are there changes in the populations of these organisms in Parkinson's disease? And does that change in the population increase inflammation? Does it increase the abnormal folding of alpha-synuclein protein, make it clump, and then it spreads? There are, believe it or not, for Parkinson's and a variety of other things, scientists have done fecal transplants, fortunately, primarily from below, but yes, and there are actually clinical trials going on to take healthy stool samples, put them in people with different disorders to normalize, make healthy the gut flora. And that's not something we're doing yet either, so don't worry. I'm not telling you all, please come back and see me. I'm not going to do all this stuff to you. But there's a lot of work being done there and evidence being found. I get asked about probiotics a lot. I don't honestly know what to tell you yet. You go on the internet and you find stuff like this where the unhappy person takes probiotics and skipping down the lane, throwing rose petals. I don't know. There have been some papers over the last year talking about certain types of bacteria that might be altered in Parkinson's, and maybe we use a probiotic that has a high population of that bacteria to normalize things. It's being worked on, but I don't know what to tell you. Nobody knows what to tell you yet. There's no probiotic that's proven to alter things yet. So I don't think that you need to go on the internet and buy all this stuff yet. I mean, it's interesting stuff. It's going to be worked out. I'd really like to have something to tell you before you do that, but I can't tell you if it will help. I can't tell you if it will hurt. We just don't know the answer to that yet. So uncharacteristically, I'm going to end like 2 minutes early because I'm a talker and that just never happens. Another is Amber Van Laar here yet? All right. Thank you, Dr. Van Laar. The two really, really cool areas of science now are how do you protect cells? Talked about it a little bit. Dr. Van Laar is going to discuss that with some specifics. How do you replace cells? People have always asked me about stem cell therapies. Why don't you just put some new cells in there? And we talked about that. Why don't you just do that? Well, there are some caveats to that based on past data in terms of, if we are right that Parkinson's disease alpha-synuclein spreads from cell to cell, if we put new cells in, are they just going to get Parkinson's disease? Now, the scientists will figure that out, but I'll let Dr. Van Laar take it from there. So this is crazy. I've never done early, and I think our talk will probably not be done early, but it'll be worth it. I would also like to thank my two helpers on this. We have two kittens who press the buttons a lot, press my buttons and press the buttons. So if there was anything misspelled up there, it's them and I didn't catch it. These guys are all over the place. But they did make it fun. So hopefully that was helpful. You should know that people are working really, really hard on this. There's a lot of good work being done. Please support the scientists. They are working really, really hard to try to figure this out so that I have better things to tell people in the clinic. We're definitely doing better now than 100 years ago, but we need to do better yet. All right, so I'm done. Thank you.